Learning objectives. At the end of this topic, you will be able to explain the importance of variations for a survival of a species, illustrate and explain modes of reproduction in single organisms, asexual. Meena and her friend Rita are on their way back home.
May I ask you a question? Why do organisms reproduce? It's a natural phenomenon. Right. After all, reproduction is not necessary to maintain the life of an individual organism. Unlike the essential life processes such as nutrition, respiration, or excretion. On the other hand, if an individual organism is going to create more individuals, a lot of its energy will be spent in the process. So why should an individual organism waste energy on a process it does not need to stay alive? So that their generation can go ahead. Right. It is obvious that we notice organisms because they reproduce. If there were to be only one non-reproducing member of a particular kind, it is doubtful that we would have noticed its existence. It is the large number of organisms belonging to a single species that bring them to our notice. How do we know that two different individual organisms belong to the same species? Usually we say this because they look similar to each other. Right. Thus, reproducing organisms create new individuals that look very much like themselves.
Let me tell you more about this in this chapter. Do organisms create exact copies of themselves? Organisms look similar because their body designs are similar. If body designs are to be similar, the blueprints for these designs should be similar. Thus, reproduction at its most basic level will involve making copies of the blueprints of body design. We have learned that the chromosomes in the nucleus of a cell contain information for inheritance of features from parents to the next generation in the form of DNA molecules. This is the information source for making proteins. If the information is changed, different proteins will be made. Different proteins will eventually lead to altered body designs. Cells use chemical reactions to build copies of their DNA. This creates two copies of the DNA in a reproducing cell, and they will need to be separated from each other. However, keeping one copy of the DNA in the original cell and simply pushing the other one would not work, because the copy pushed out would not have any organized cellular structure for maintaining life processes. Therefore, DNA copying is accompanied by the creation of an additional cellular apparatus and then the DNA copies separate. Effectively, a cell divides to give rise to two cells. No biochemical reaction is absolutely reliable. Therefore, it is only to be expected that the process of copying the DNA will have some variations each time. As a result, the DNA copies generated will be similar but may not be identical to the original. Some of these variations might be so drastic that the new DNA copy cannot work with the cellular apparatus it inherits. Such a newborn cell will simply die. On the other hand, there could still be many other variations in the DNA copies that would not lead to such a drastic outcome. Thus, the surviving cells are similar to, but subtly different from each other. The importance of variation. The consistency of DNA copying during reproduction is important for the maintenance of body design features that allow the organism to use that particular niche. Reproduction is therefore linked to the stability of population of species. However, niches can change because of reasons beyond the control of the organism. If a population of reproducing organisms was suited to a particular niche, and if the niche were drastically altered, the population could be wiped out. However, if some variations were to be present in a few individuals in these populations, there would be some chance for them to survive. Thus, if there were a population of bacteria living in temperate waters, and if the water temperature were to be increased by global warming, most of these bacteria would die, but the few variant resistant to heat would survive and grow further. Variation is thus useful for the survival of species over time. Modes of reproduction used by single organisms. The modes by which various organisms reproduce depend on the body design of the organism. Basically, there are 6 modes of reproduction by single organisms. They are fission, fragmentation, regeneration, budding, vegetative propagation, and spore formation. Let's know about fission. For unicellular organisms, cell division of fission leads to the creation of new individuals. Many different patterns of fission have been observed. Many bacteria and protozoa simply split into 2 equal halves during cell division. In organisms such as amoeba, the splitting of the 2 cells during division can take place in any plane. However, some unicellular organisms show somewhat more organization of their bodies, such as is seen in Leishmania, which causes Kala-Azar, which have a whip-like structure at one end of the cell. In such organisms, a binary fission occurs in a definite orientation in relation to these structures. Other single-celled organisms, such as the malarial parasite, Plasmodium, divide into many daughter cells simultaneously by multiple fissions. Yeast, on the other hand, can put out small buds that separate and grow further. Regeneration. Many fully differentiated organisms have the ability to give rise to new individual organisms from their body parts. That is, if the individual is somehow cut or broken up into many pieces, many of these pieces grow into separate individuals. For example, simple animals like Hydra and Planaria can be cut into any number of pieces, and each piece grows into a complete organism. This is known as regeneration. Regeneration is carried out by specialized cells. These cells proliferate and make large number of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. These changes take place in an organized sequence referred to as development. However, regeneration is not the same as reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. Budding. Organisms such as Hydra use regenerative cells for reproduction in the process of budding. In Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals and, when fully mature, detach from the parent body and become new independent individuals. Fragmentation. In multicellular organisms with relatively simple body organizations, simple reproductive methods can still work. Spirogyra, for example, simply breaks up into smaller pieces upon maturation. These pieces of fragments grow into new individuals. This is not true for all multicellular organisms. They cannot simply divide cell by cell. The reason is that many multicellular organisms are not simply a random collection of cells. A basic strategy used in multicellular organisms is that different cell types perform different specialized functions. Following this general pattern, reproduction in such organisms is also the function of a specific cell type. There must be a single cell type in the organism that is capable of growing, proliferating, and making other cell types under the right circumstances. Spore formation. Even in many simple multicellular organisms, specific reproductive parts can be identified. The thread-like structures that developed on the bread are not reproductive parts. On the other hand, the tiny blob on a stick structures are involved in reproduction. The blobs are sporangia, which contain cells or spores that can eventually develop into new rhizopus individuals. The spores are covered by thick walls that protect them until they come into contact with another moist surface and can begin to grow. Vegetative propagation. There are many plants in which parts like the root, stem, and leaves develop into new plants under appropriate conditions. This property of vegetative propagation is used in methods such as layering or grafting to grow many plants like sugarcane, roses, or grapes for agricultural purposes. Plants raised by vegetative propagation can bear fruits and flowers earlier than those produced from seeds. Such methods also make possible the propagation of plants such as banana, orange, rose, and jasmine that have lost the capacity to produce seeds. Another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parent plant to have all its characteristics. Buds produced in the notches along the leaf margin of bryophyllum fall on the soil and develop into new plants. Summary. Let's summarize the topic. Today, we have learned that reproduction involves formation of a DNA copy and additional cellular apparatus involved in the process. Depending on their body design, various organisms use different modes of reproduction. Vegetative propagation, plants can bear fruits and flowers prior to those produced from the seeds. In fission, many bacteria and protozoa simply divide into 2 or more daughter cells.